Hi there, and welcome to this video on A-Level Biology for the AQA specification, focusing on the topic of immunity, and in particular, on the types of immunity and vaccination. I'm Manisha from StudyMind, where we help you to revise A-Level Biology with our helpful video tutorials tailored to your subject, your specification, and to you. If you're new here, please make sure to click that subscribe button. And whilst you're watching, feel free to leave any comments down below of anything you're unsure about, and let us know if it's your first time watching so we can send you our free revision resources. We also have helpful timestamps to guide you through the specification. So, let's get started. Welcome to lesson 5 of 6 in this tutorial, covering the types of immunity and vaccination. This is the 5th video in our series of 6 lessons on the topic of immunity. In the last lesson, we looked at the structure and function of antibodies. Here are the key learning objectives for today's lesson. First, we'll be looking at active and passive immunity, then at herd immunity. We'll then move on to the ethical considerations of antibodies, and finally at antigenetic variability. Here are the AQA specification points for today's lesson. Feel free to pause the video now and have a quick read through them before we begin. We'll start off by looking at the differences in active and passive immunity. There are 2 types of immunity, active and passive. Active immunity is developed when the body makes its own antibodies in response to pathogen exposure. Active immunity is due to memory B cells and antibodies, which are typically long-lasting. An individual may need to obtain booster vaccinations after a period of time in order to keep their immunity. Sometimes, the immunological memory fades after a certain amount of time. Passive immunity is from an injection of antibodies from another animal. In many cases, individuals infected by a particular pathogen need immediate treatment. One way of doing so is by injecting antibodies produced in an animal or antibodies from an immune person into the infected individual. Once injected, the antibodies can quickly identify the pathogen and eliminate the infection. Here's a good summary of active and passive immunity. Now, we'll look at the use of vaccines in herd immunity. Our body can itself develop immunity, given enough time. Upon infection, given enough time, our bodies can gain active immunity against a pathogen by developing an immune response. However, this usually takes 2 weeks. Many pathogens act way too quickly for our immune system. The process of mounting a successful primary immune response and gaining immunological memory usually takes around 2 weeks. Many pathogens, such as the Ebola virus, can kill an individual in a matter of days. Vaccines enable the immune response to occur quickly. Vaccination allows the body to be fully prepared to effectively combat pathogens. The peak of the immune response occurs within the first 3 days. Now let's look at herd immunity. Mass vaccination contributes to herd immunity. This means that the virus essentially disappears as it cannot pass from person to person. This means that members of the population, such as infants, who have not been vaccinated, are protected against the pathogen. This phenomenon is known as herd immunity. Many infants are too young to receive particular vaccines. Most vaccines are given to an individual over different stages of childhood in order to prevent certain risks. Therefore, strong herd immunity is necessary in order to protect infants and other individuals who can't be vaccinated for various reasons. Now let's look at some ethical issues associated with the use of vaccines. If individuals in the population begin to lose their immunity by not vaccinating themselves or their children, it leads to the loss of herd immunity. Loss of herd immunity leads to the resurgence of a disease. Non-eradicated diseases can return. Diseases such as whooping cough and MRI were almost eradicated due to vaccines. However, concerns over vaccinations led to a big fall in vaccinations for these diseases. This led to the rapid resurgence of both of these conditions, which can be fatal to infants and young children. Here are some of the risks of vaccines and their counter-arguments. Feel free to pause the video now and have a read through them, but remember, overall there is no evidence that vaccines pose a risk to society. Animals may be given diseases to stimulate antibody production, and there are concerns over animal rights. Often, mice are given induced cancer. Side effects may occur for humans, such as allergic reactions. Monoclonal antibodies have been used to treat multiple sclerosis, but this has unfortunately led to some deaths. However, vaccinations can be used to treat many conditions, so saves money and time for the NHS. They can also be used to detect early pregnancies in order to help mothers. And monoclonal antibodies as a cancer treatment have less side effects than alternative therapy. They are specific and therefore useful in diagnosing diseases quickly. Can you think of all the positives and negatives of vaccines? Now, let's look at the definition of an antigen. A vaccine stimulates the primary immune response, producing memory cells against a specific antigen. It assumes that if there is a second infection, then the same antigen will be present on the pathogen. Sometimes pathogens, especially viruses, will evolve and alter their antigens, leading to antigenetic variation. When these pathogens infect a second time, the memory cells are ineffective and a new primary immune response needs to be generated. Antigenic variation makes vaccination difficult. If the antigen changes, then the vaccine will lose its effectiveness. There are two types of antigenetic variation. One is antigenetic drift, and the other is antigenetic shift. Antigenetic drift involves minor variations in the antigen structure. This results in the production of new strains of a particular virus. Antigenetic shift involves major variations in the structure, usually leading to the development of entirely new types of viruses. We've now covered all the learning objectives for today's lesson. If there's anything you feel unsure about, feel free to skip back through the video and re-watch it. We've now covered Lesson 5. If you enjoyed this tutorial, make sure to subscribe by clicking down below and leaving a comment of the topic that you'd like to see a video on. Click here to watch the rest of our videos in our A-Level Biology series, or visit our website, studymind.co.uk, for past paper compilations by topic and specification.